Okay, everybody, welcome to lecture 2. Lectures are a bit interesting because it seems like they kind of repeat some information. So, um, not going to cover all the slides. If I've already done the slide on lecture 1, I'm not going to go over it again. But yeah, um, all this stuff. Um, Yeah, so just real quick, going over mechanism for translational control. Um, this is an interesting section just because of how it's formatted. But this is basically talking about the guanosine cap, essentially, I believe. You guys have already seen this in 441, so I'm not going to harp on it too much. Um, so, yeah, basically the idea is that, when you make an mRNA, there's a 5' phosphate or 5' triphosphate. And basically, one of the enzymes removes one of these phosphates, and then another one called guanyl transferase adds a GMP to the end, and it creates a weird triphosphate linkage. Again, you guys saw this in 441. And then there's a methyl transferase enzyme that adds a methyl group to the guanosine to make it a 7-methyl guanosine. But yeah, but basically all that does is it basically functions as a protector, so it protects mRNA from degradation. So the things I probably memorize from this are, a phosphatase. So basically this section here, the fact that, you add a 5' triphosphate linkage, or you have a 5' triphosphate, use a phosphatase, then use a guanyl transferase to add a GMP, and then use a methyl transferase to add the methyls that protects it. So here we've got this section is a bit interesting because it's kind of I believe it's supposed to be the section talking about like the poly A tail, essentially. So up here we talked about the 5' cap, but basically what happens here is, when you're making the poly A tail, you bind 3 proteins to this synthesized mRNA. You cleave the mRNA at this GU/U rich region. It's degraded in the nucleus, and then what you do is you do there's a poly A polymerase that adds 200 nucleotides to the end. It doesn't require a template of any kind. So basically, we're just trying to show you how you make the poly A tail and how you make this the 5' cap. So yeah, and the function of this is also to protect it from degradation. So yeah. So these mRNA modifications, so the 5' cap poly A tail, they allow the RNA to take a closed loop structure as shown up here. And this is achieved with the help of a couple proteins. So you've got EIF4E and EIF4G. So EIF4E binds the cap, this guy, and then EIF4G is a scaffold that binds to EIF4E, and it also binds to the polyadenylate binding proteins, which bind to the poly A tail, which leads to that kind of circular formation. So this closed loop kind of improves translation efficiency because the mRNA is stabilized, it's not going to degrade. Basically, it just increases the rate of initiation and translation. So this is just something you have to memorize, I'm going to be honest with you. Like EIF4E and EIF4G. You guys get a note card, I believe, if I'm not mistaken, so put this on your note card. It's going to be helpful to know. So for this, what are the mechanisms of translational control? What are the factors through which translation can be modulated? There's a bunch of ways you can do it, right? So you can, anything that modifies the 5' cap process, the poly A tail, the EIFs, the EIF2s, amino acids, tRNAs, there's a whole bunch of ways you can change this. So basically, you can modify initiation, modify access to the mRNA, and then modify stability. So example of translational control, we're going to talk about this. So under what condition would you like to globally decrease protein synthesis? An example of this would be, starvation, viral infection, and heat. So EIF2 is frequently modified to modulate the levels of global protein, basically. Because remember, if you phosphorylate EIF2, it stops it from exchanging the GDP for GTP, because the EIF2B binds and gets stuck, right? So again, talked about this. EIF2 is actually a trimeric G protein, so it's like got like an alpha, beta, gamma, if you will, subunit. So when EIF2 is active, it's bound to GTP, and then it binds to the tRNA, right, this thing. And this complex can bind to the small ribosomal subunit, which allows it to act as a flag for the large ribosomal subunit. So once this complex is built, the small ribosomal subunit can then bind this massive complex through the 5' cap. And, it also is assisted with the EIF4E and EIF4G. And then, it uses the small ribosomal subunit, then uses ATP to kind of move along the mRNA until it finds the AUG sequence. And then once this happens, you hydrolyze the GTP. This gets stuck onto the ribosomal small subunit, and then the large subunit slaps on top. And so I talked about this before, but I didn't really show it here very well. But you can imagine, this is the rest of the process. So, like I said, after this hydrolyzes, the large ribosomal subunit slaps on, and then you continue with translation like normal. But, like I said before, EF2 is bound to GDP, and then it requires this. So after hydrolyzation, it's bound to GDP, and it requires EF2B to release the GDP for a GTP, and so that's where you get the release from. So the phosphorylation of EIF2 regulates protein synthesis through this process like we talked about. I feel like I've repeated this like 8 times. But again, this is what it's supposed to look like normally, where you have EIF2, EIF2B binds to this thing, causes the release of GDP, end up with this, then you attach the GTP, and that causes dissociation, so this separates from that. But under phosphorylated conditions, the EIF2 is phosphorylated by this, phosphorylated by a kinase, protein kinase, and even if EIF2B binds to it to try and switch out the GDP for a GTP, it still doesn't work. So, all that to say, this. So, what happens if there's a mutation in EIF2B? Will translation increase or decrease? Well, if you mutate EIF2B and it breaks, then you're not going to be able to do this process. So basically this step doesn't happen. So you're stuck in this form. So you decrease translation, which is why this is the answer here. And so let's say you generate a phosphomimetic mimic of EIF2. So what that means is that there is, it always looks like it's phosphorylated. So it always looks like this. Will translation increase or decrease? It'll decrease, because remember, even EIF2B, it'll still bind, but it'll end up in this stuck form, so you're going to sequester all of your EIF2B. And so you never get any GTP form. You never get this form. Talked about this already. Don't think it's too important to go over this again, but basically, like I talked about before, endonucleases, you guys have talked about endonucleases, right? They cleave DNAs and mRNAs and all that stuff from the inside out, and the opposite of that is an exonuclease. Exonucleases start from the outside and go in. But yeah, here, eternally, endonuclease can cleave mRNA internally, thus decapping one half and removing the poly A tail from the other. So here you've got a case where you're breaking off the ends here, and then we have two options. You either degrade 3 to 5, so you degrade the poly A tail, or you decap and then degrade this way. So depends on how you feel, I suppose. Deadenylases gradually shorten the poly A tail. We talked about this before. This shortening process occurs because of deadenylases, generally speaking. Remember that this complex is in this weird equilibrium with the deadenylase version, where the deadenylase is bound to the 5' region, and then once it's bound to the 5' cap, it eats away at this poly A tail, right? So yeah, there are two post-translational controls mediated by iron. We already talked about this a little bit. So iron is necessary for all cells, pretty much. Very important in the electron transport chain. Remember, you guys talked about this actually in 440 some of the complexes use iron, and also important for ATP synthesis in the mitochondria. So aconitase, which is what we're going to talk about later, actually uses iron in its stuff. So iron deficiency, anemia, iron excess is hemochromatosis. So yeah, you can't have too much or too little iron. Talked about ferritin, talked about transferrin. So you guys are going a little bit more in depth into these a little bit. So ferritin is a globular protein. It stores iron inside cells, and it kind of acts as like a storage system, similar to how glycogen is like a storage of glucose. That's what ferritin is. It's a protein that stores. It's a protein specifically that stores iron in it. And then transferrin is a transporter for iron. It's like a transport channel, if you will. So now we have to think about a couple of things. So ferritin is for storing iron. So, if you're under iron-starved conditions, you're probably not going to want a lot of ferritin because you don't want to store it. You want to be having it. So it's probably going to be less ferritin, and transferrin is going to be more of it because you want to make sure you're transferring more iron into your cells. So iron in excess, you're going to want more ferritin and less transferrin, right? So there you go. Yeah, okay, I was like really confused how this was formatted. Hopefully this makes sense. Ferritin, you don't want to store iron if you need iron, right? It's like the whole thing with glycogen and glucose. You don't want to store glycogen if you need to have glucose for your muscle cells. You're going to break down the glycogen, so you don't want to store it, you want to use it. So less ferritin because less storage, more transferrin because you want more active iron in your cells. If you have excess iron, you're going to want more ferritin to store it, and then you're going to want less transferrin because you don't want to uptake more iron than you need. So, talked about these. Aconitase is the main driver of, how these things are translated and regulated. I'm not going to talk about this again. The basic gist of this is under iron-starved conditions, the aconitase blocks translation of ferritin, and then it promotes transferrin production. Whereas in excess iron conditions, iron binding to aconitase leads to release of the aconitase from the transferrin mRNA, which causes it to be degraded, and then also causes the release from the ferritin mRNA, which causes it to be made. So, here we're going to talk about, microRNAs. What are microRNAs? What do they do? There's a bunch of microRNAs. All you need to know about a microRNA is that, there's tens of thousands of different microRNAs. What they do is that they complementarily bind to mRNAs, and they kind of help either fine-tune their translation or degrade them. Basically, they modify translation rates. And, they're pretty short. About 23 nucleotides long, much shorter than a lot of mRNAs and obviously DNA. So I talked about this already. Here's a RISC complex. So this is one way. Remember, I talked about this before, like this pathway here. So remember, maybe you transcribe an RNA and then you crop it in the nucleus, then you dice it into smaller sections of 23 bases, around 23 bases per microRNA. Then you bind these little sections to these argonauts and other proteins. Oops, sorry about that. And when you bind them, you form these RISC complexes. And I can't remember what RISC stands for. I think it stands for RNA-induced silencing complex, but I can't remember off the top of my head. It's not that important, to be honest with you. Let me see if I can find the... I think it is called RNA. Yes, it is an RNA-induced silencing complex. Okay, I had it right. Okay, cool. Right, so... like I was saying... yeah, so, you do this, you dice it, then you attach it to argonauts, and you get these RISC complexes. And it kind of shows you here, so... microRNAs are synthesized by RNA polymerase II. They're capped polydenylated, then... through complementary base pairing, it becomes double-stranded. It's cropped in the nucleus, exported to the cytosol, where it is diced by an enzyme, dicer, and then argonaut and the RISC complex associate with both strands and then cleave and discards one of them. So basically you form this thing. Basically the Argonaut binds to this thing and it cleaves one of them, and so it cleaves one and disposes of it, and then it grabs the other one and makes it part of the complex, basically. And so we talked about this before, where now you're here. If there is a little bit of base pairing, it leads to translational repression, so it doesn't slice the mRNA. So yeah, this is what we're talking about after the Argonaut binds. So if the RISC complex binds to an mRNA weakly, it doesn't cleave it. Instead, the mRNA is repressed. So it repressed, and then eventually it does degrade. But then if it's an extensive match, the mRNA is cleaved by the Argonaut, and you remove the mRNA poly-A tail, which causes it to be exposed to exonucleases, and you break it down after. So I talked about this already. They can have multiple targets. They can act at the same time on one mRNA, and they can also be cell specific, right? So liver cells. So like I talked about glucose 6-phosphatase. You don't want glucose 6-phosphatase in the brain, or actually I guess this is a bad example. Glucose 6-phosphatase is not the best answer. What would you want in the brain? Microtubules. So I guess so microtubules are very, very prevalent in neurons. I think a lot more prevalent in neurons than they are in some other cells, just because of how long neurons can become compared to liver cells. So maybe, obviously liver cells need microtubules, but they don't need them as much as brain cells probably. So for example, if you have a brain cell, you don't want to make a microRNA for that in that cell, while for liver cells you would, because you don't want as much expression of microtubules. So you can see here in the brain, no microRNA leads to lots of expression of microtubules, which is great because you need them for structure. But in liver cells, you do have a microRNA because you don't need as much of them. So here we're going to talk about cancer a little bit. So you can see here scenario 1, protein prevents cancer. So protein A prevents cancer conditions. So a healthy cell should have a lot of protein A. Makes sense. It has very little microRNA, a lot of protein A. In a cancerous cell, though, maybe the cancerous cell has developed a microRNA, or a surplus way of, or a excess way of producing these microRNAs that inhibit production of protein A. So too much microRNAs, too little A proteins. So because there's too little A proteins, A is probably a tumor suppressor. So too little A, which leads to cancer. So what kind of function does protein A might have? It probably blocks cell cycle. It acts as a tumor suppressor. See exactly, yep. Protein A could be a I don't know about this. Protein A could be a proto-oncogene or involved in transcript. This is true. It could be a transcriptional proofreading thing. Proto-oncogene is a... I don't know about that. That seems a bit... doesn't seem right to me. I would double check that. That's... because, like, in this case, you have to remember that more of this doesn't lead to cancer, which means that it's actually better to have this, so it's clearly suppressing undivided cell growth. But in this case here, we have cancer is actually caused by less A, which means that, less tumor suppression leads to cancer, which to me sounds like this should be a tumor suppressor and not a proto-oncogene. So I would double check that with your professor, if they didn't already clarify it. So yeah. Protein B is the opposite here. So if we see healthy cells, they don't have a lot of B, but cancer cells have a lot of B. So that means cancer, so this is probably acting as a proto-oncogene, probably promotes cell growth and division. So the cancer cell has found a way of repressing this microRNA from being produced. So yeah. So, how can you use microRNAs? MicroRNAs can be useful targets for cancer biomarkers. and integumeres. So the integumeres are basically microRNAs that bind to other microRNAs. So they soak up or sequester microRNAs, and they can be used in combination with chemotherapy to target proteins, all that stuff. So remember, here we have a, protein A prevents cancerous activity, and the microRNA is a biomarker indicating cancer. So here, yes. So here, if you look at this, the healthy cell has a, I wish they didn't show it like this. That's okay. The healthy cell has a lot of A, right? And the cancer cell has a lot of this microRNA. So what you do is you make an intagomere, and this intagomere binds even more strongly to the microRNA than the strength of the microRNA binding to the mRNA. So basically, the microRNA to intagomere binding strength is stronger than the microRNA to mRNA binding strength. And so basically what happens is the microRNA ends up binding to the intagomere more often, which means that the microRNA cannot repress the translation of A anymore, and so A is now going to be translated, and so you don't get cancer, or cancer is mitigated. Whereas for scenario 2, maybe protein B promotes cancerous activity, and actually, yeah, this actually further proves what I was saying upstairs. Protein A prevents cancerous activity. It shouldn't be a proto-oncogene, I don't think. Should be a tumor suppressor. Anyways, as I was saying, protein B promotes cancerous conditions. So in that case, the absence of the microRNA is the biomarker for cancer. So all you have to do then is add microRNAs to stop B being made. Cool. This right here is, microRNAs in cancer, just an example. So cancer cells injected into the liver of these mice. After 4 weeks, 14 days without microRNA 434, you measure the size. So they have a lot of tumors. They have a cancer. So basically, you inject these cancer cells, you don't do anything to them, they develop cancer in 14 days. If you inject them with cancer, then add the microRNA, it actually prevents cancer, or at least it slows it down significantly. So is it being expected to prevent or promote cancer? I know it tells you the answer here, but it clearly is preventing cancer. If you leave the cancer cells alone and you don't do anything and it causes severe cancer, and then if you add a microRNA and it stops the cancer, that means that we're clearly negating the cancer or preventing cancer. And so you'd probably expect that, maybe this microRNA is probably blocking the production of a not a tumor suppressor, production of a proto-oncogene or something. It's clearly stopping translation of some sort of cell cycle promoting protein or thing. So here's a different example. So cancer is injected into these mice. 2 days, 3 weeks, have quite large cancer cells. But then if we look at this, you inject cancer cells, you add this intagomere, and then it leads to improvement. So the fact that the intagomere being added leads to better cancer means that this initial microRNA is probably binding to a tumor suppressor mRNA. So by adding the intagomere soaks up the microRNA, and the tumor suppressor can be translated, and so you lead to less cancer. So the microRNA 10 is promoting cancer. Remember, because the microRNA is blocking a tumor suppressor. So it promotes cancer. So, here they give you, more examples of this kind of stuff. They talk about microRNAs and cholesterol. So this microRNA leads to production of cholesterol, which leads to more serum cholesterol. So when you add the intagomere, what should happen is you actually make less of this. You should drop levels of this. You should drop levels of cholesterol, which is good. But yeah, if you have a mutant Western diet, if you add a mutant, basically what happens is you get large plaques. But then if you add the intagomere, again, like we said, if you add the integument blocks this drops significantly, which leads to less serum cholesterol. So integumers are very useful. So how do you make microRNAs for research? Couple ways. So plasmid or viral vectors. So shRNAs are small hairpin RNAs, which is why they're called SH, small hairpin. So basically, you add a plasmid, you have a promoter, you have this designated sequence that you want to become an mRNA and eventually a silencing RNA or small interfering RNA. And so once this is transcribed, it makes this hairpin structure, which is processed by dicer into this thing, which is then processed by RISC, which then, after being processed by RISC, is turned into some sort of microRNA that can now target mRNA. The other option is that you just buy small interfering RNAs from a company. So you can either make them yourself using viral vectors and the cell machinery. So make it yourself, make a promoter mRNA yourself, have it be diced up, processed by RISC, turn into a microRNA, or just buy the microRNA, or buy the small interfering RNA directly from, just buy it from a company of some sort, and you basically just end up with this step immediately. Still has to be processed by RISC though, but eventually it does act as a silencing RNA or a microRNA, a small interfering RNA, whatever you want to call it. Okay, so poliovirus. Polio is interesting. Yeah, I mean, polio is bad. It causes paralysis, destroys nerve cells. The spinal cord gets absolutely destroyed by poliovirus. So, deadly virus, horrible. Basically, we talked about this, but under normal conditions, you have this. In eukaryotes, we have this machinery that is used for translation. And so this is kind of what it looks like normally. So normal mRNA translational control uncapping by poliovirus. So in a normal cell, you just do this. You have your EIF4G, your EIF4E, and then you bind your small ribosome subunit, your methionine, and everything's great. But in a polio-infected cell, there is a polio enzyme that ends up cleaving EIF4G, and the cleavage of that leads to this production of this 4G thing. And so what ends up happening is, the poliovirus or the polio-infected cells, they have polio mRNAs that were made from its own genetic data. And those polio mRNAs have a internal ribosome entry sequence. Basically, and what happens is after you've cleaved this EIF4G using the polio enzymes, that portion of the EIF4G binds to these entry sites, or these ribosome binding sites, and allows for the recruitment of ribosomes to the polio mRNA specifically. So not only are you breaking the machinery for the normal translation process, you're also increasing the ability for the polio mRNAs to get translated, because once you've cut the EIF4G and split it, that part that you cut off is able to bind to the ribosome binding sites of the polio mRNAs and causes recruitment of ribosomes to those mRNAs as opposed to the normal mRNAs of your cell. So this is the danger with the polio virus. Hopefully that makes sense. So, remember, polio is its own virus. It has its own genetic data. So it uses its genetic data to make these mRNAs. It just so happens that one of the enzymes it produces furthers its goal in producing more of its mRNAs and producing more of its proteins. So translational control, endogenous use of internal ribosome entry sequences. Yeah. This is there. There's really not much to say about this. The cells sometimes use their own internal ribosomal entry sequences. But I don't think this is something that's too crazy. Something to keep in mind for sure, but I don't think it's that crazy. Oh yeah, this is an interesting. So translational control. So other uses of IRES sequences. So yeah. So basically, this is saying like under stress conditions, these IRES sequences actually allow for translation, even when maybe EIF2 is phosphorylated, right? And you can't really translate using that mechanism. So for example, P53, cancer preventing gene, very important, one of the most important cancer prevention genes. This uses an IRES, so it's not super dependent on things like EIF2 and things like that. So even under stress conditions, these things can still function very well. The other thing is that you can use them for multiple protein expressions. So you can use them to not only translate, this ORF here, but you can also use it to recruit another ribosome to this site. Because remember, ribosomes can bind to the same mRNA at the same time. They don't have to bind one at a time. So maybe you bind one ribosome here, you bind another ribosome here to this IRES, and then you produce both this ORF and this GFP. So it's a good way of being able to measure it. So because if you measure production of GFP, it most likely means you're also producing ORF1. So here is actually an example of that. Yep, see, look. So translational control, IRES sequence for reporter gene expression is exactly what I just talked about. So you have a target gene with a plasmid, you transfect it, you have no idea which of these cells have this gene that you're looking for. However, if you input the target gene and you input a GFP with the IRES, now you do know which cells which cells for sure got the plasmid, right? Because you're able to say, oh, okay, both of these genes are present within my cell, and it's definitely gonna express the GFP, because the GFP is attached to an IRES, so it's definitely gonna express this. So now it's gonna start glowing when it is transfected with this plasmid, and you can see that here. So now you can tell which cells have the plasmid that you're interested in. Okay, now we're going to switch gears a little bit. We're going to talk about protein trafficking. So we're going to talk about how they find their compartments, budding, fusion of vesicles, ARF, clathrin, RABs, snares, all this stuff. But yeah, there's a whole bunch of stuff to talk about here. So I'm not going to go over all this. This is not super necessary. There's just a lot of random stuff here that's not super important. This is a very simple way of talking about. So there's two ways of moving proteins across hydrophobic membranes. You can either use translocation using a protein, or you can use vesicles. That's it. So you can see here, this is a really poor example actually, but so there's actually a channel that should be shown here, but basically a protein can be fed through a channel, and that's one way of translating. It's not really shown super well here at all. Or you can use vesicles, right? So vesicles are like these membrane phospholipid circles. So you can see how it forms here. So you can see how it's budding here, these little proteins inside, and then it buds, and then it fuses again, and then it releases. So those are two ways of transporting proteins. So again, you show this. We have this, like whatever this phospholipid thing is. We have a budding event happening. Budding, it pinches off here, and then this thing fuses with this next membrane. So here is kind of like the way, yeah, okay, that's actually really important. So protein domains that start off inside the ER lumen end up outside the cell. So notice, this is inside the ER, this like circle. Notice how it ends up on the outside of the cell, right? Very important to remember. Please remember that. Super important. So basically, the portion of the protein that is touching the cytosol in the beginning also touches the cytosol in the end. So this flat part in the ER, this protein is in the ER membrane, right? Flat part is in the cytosol. Once it's been transported to the Golgi and then eventually to the cell membrane, it's still the flat part in the cytosol. True for all proteins, pretty much. Some exceptions, of course, but always true. Please remember that. For this, protein trafficking, budding, and fusion, specialized coat proteins create membrane buds. This is just a way of talking about there are certain proteins that cause the membrane to bud. So you can see here budding starts because of these green proteins. And so then you have this coated vesicle. This vesicle is then uncoated, and then this thing can fuse normally. So it fuses because of target recognition first, and then it fuses properly. So, yeah. So, there's two types of transport. There's retrograde transport and anterograde transport. Retrograde transport is going from the plasma membrane back to the ER/Golgi, and anterograde transport is going from the ER to the membrane, basically. Cool. There are many different types of proteins that help with these transport processes, so retrograde and anterograde. They are called clathrin, COP1, and COP2. It's like police, basically. I think of COP1 and COP2 as like the policemen of the transport system. They basically make sure that things are in order properly. So clathrin is a really weird protein. It looks like this, which I don't even know what to think of that. It just looks weird. It doesn't look real. But it's a cool little protein. But yeah. Basically it just helps with the process of transporting proteins and protecting them. Here's just an example of COP2 and stuff. I'm not gonna go over this video. But basically all you need to know is see this so basically COP2, if I'm not mistaken, I think it might be covered in the next video. COP2 basically, if I'm not mistaken, is anterograde transport, generally speaking, and then COP1 is usually retrograde transport. Don't quote me on that. I think I'll cover it in the next video if I'm not mistaken. We'll see. But basically, just like, these like vesicle coating thingies, that's basically what COP1 and COP2 are. They help with the transportation process from ER to membrane or membrane to ER. So that's what we covered today. That's actually the end of this video. It's actually a longer video today, and my mouth is killing me. My mouth is really dry. But I hope this video was helpful for you guys. I know I'm going through content pretty quickly, but I'm trying to get these out for you, so yeah. Other than that, hope you have a good day, and happy studying, guys.